Thin layer chromatography. Thin layer chromatography is also called as TLC. It is a technique used to isolate non-volatile mixtures. The experiment is conducted on a sheet of aluminum foil, plastic, or glass which is coated with a thin layer of adsorbent material. The material usually used is aluminum oxide, cellulose, or silica gel. On completion of the separation, each component appears as spots separated vertically. Each spot has a retention factor RF expressed as RF is equals to distance travelled by sample divided by distance travelled by solvent. The factors affecting retardation factor are the solvent system, amount of material spotted, absorbent, and temperature. TLC is one of the fastest, least expensive, simplest, and easiest chromatography technique. Principle: TLC depends on the separation principle. The separation relies on the relative affinity of the compounds towards both the phases. The compounds in the mobile phase move over the surface of the stationary phase. The movement occurs in such a way that the compounds which have a high affinity to the stationary phase move slowly while the other compounds travel fast. Therefore, the separation of the mixture is attained. On completion of the separation process, the individual components from the mixture appear as spots at respective levels on the plates. Their character and nature are identified by suitable detection techniques. Procedure: before starting the TLC experiment, it is important to understand the different components required to conduct the procedure along with the phases involved. Firstly, thin layer chromatography plates. Ready-made plates are used which are chemically inert and stable. The stationary phase is applied on the surface of the plate as thin layer. The stationary phase has a fine particle size and a uniform thickness, ensuring accurate separation of components. TLC chamber. The chamber is used to develop the TLC plates. It provides a steady environment inside, essential for proper development of spots. The chamber helps to prevent solvent evaporation and keeps the process dust free. Thin layer chromatography mobile phase. The mobile phase refers to the solvent or the solvent mixture that moves up the plate. It must be free of particulates to avoid interference in the chromatographic process. A high purity mobile phase results in better spot development and more accurate results. TLC filter paper. A filter paper is placed inside the chamber and is moistened with the mobile phase. It helps to saturate the chamber with solvent vapors, ensuring uniform movement of mobile phases and enhanced resolution of spots. Applications: it is used in identifying compounds in medicines, in detecting adulterants in herbal products, in monitoring chemical reactions, in analyzing amino acids and peptides, in testing purity of chemicals, detecting pesticides in food, studying dyes and pigments, analyzing plant extracts, in forensic ink analysis, in control of perfumes and essential oils, detecting counterfeit drugs, in separating lipids, in identifying food additives, in toxicology screening, in verifying chemical identity, in separating small organic molecules, in detecting industrial contaminants, monitoring chemical degradation, in analyzing steroids and hormones, and in teaching chromatography principles. Thank you.